There are three types of neurons in the nervous system, and they're all sort of defined by the amount of dendrites they have. And again, dendrites and axons are both called cellular processes. The first group is the most common. It's called a multipolar neuron. It has many processes extending from the cell body, so you can see lots and lots of dendrites there. It has many receptive regions because of those dendrites, and it's the most abundant in the body. We can see the trigger zone here, the axon, and then here's the secretory region where neurotransmitters are going to be released. The second type of neuron is a bipolar neuron, where there are two processes, a dendrite and an axon. These do not generate action potentials and are rare and only found in the special senses, like the olfactory mucosa, the eye, and the ear. The third type is a unipolar or pseudo unipolar, because it looks like there's only one process, but there's really, part of it's kind of like a dendrite, part of it's kind of like an axon, but it's one process that extends from the cell. And we can see that axon shown here. These are the receptive endings where the dendrites kind of merge to the axon, but really it's one process, or looks like one process. It's mainly in the peripheral nervous system, and it's common only in the dorsal root ganglia, which is going to be in chapter 13, the peripheral nervous system. When we look at the other characteristics of these, we can see the multipolar, the most common, is going to have many dendrites, one cell body, and one axon. And a place we could find it is in the cerebellum and what are called the pyramidal cells. You don't need to know those terms, but they're kind of found in the frontal lobe, and they extend to the body. We'll have a more specific name for that when we get to chapter 12. The bipolar neurons, again, are only going to have two processes, a dendrite cell body and then an axon. So we could see that in the olfactory cell for smell and the retinal cell for eye. And then finally, the unipolar is going to have the receptive ends basically leading to the axon, a cell body, and we find it in what's called the dorsal root ganglion. When we further compare those, we see the rest of that table that's in your textbook. The multipolar are called interneurons because they conduct impulses within the central nervous system. So they integrate sensory or motor output. So some multipolar neurons are also motor neurons because they conduct impulses along the efferent pathway to a muscle or gland. So you can see an example of an interneuron here and a motor neuron that's going to transmit that action potential very quickly to the muscle in this case. And these arrows represent action potentials. Bipolar neurons, however, are going to be sensory neurons in some special sense organs. So in the retina, we see a bipolar neuron of the retina that transmits that impulse to the brain from the eye. And finally, the unipolar ones are also sensory neurons. They conduct afferent pathways to the central neuron for interpretation. So this would be an example of taking an afferent impulse from the skin via a sensory neuron to the spinal cord. And this region right here is what identifies it as a dorsal root ganglion. And you will remember from a couple slides ago that when we see the word ganglion, we need to remember that that's in the peripheral nervous system. So we're going to start out talking about the resting membrane potential before we get to graded potentials and action potentials. When they first made the discovery, they made this discovery on a squid which has a super long axon. So what they did was they put a microelectrode inside the cell connected to a voltometer. And they discovered that the plasma membrane literally acted as like a battery, in that there is one end that's positive and one end that's negative. So that's how I think it's helpful to think of the plasma membrane. There's an outside that's positive and an inside that is negative. So this would be the extracellular fluid, and this would be the intracellular fluid. And the difference between it would be negative 70 millivolts. And that means that the membrane is polarized. So there's a difference between the inside and the outside due to charges as well as chemicals. So it leads to an electrochemical gradient. There's a difference in charges, which you can clearly see. Also, there's a difference in ions. There's sodium ions, chloride ions, potassium ions on the inside, as well as amino acids, which are negatively charged. But the difference between the outside means that the inside has a charge of negative 70 compared to the outside. So there's this potential difference that exists across the membrane. And that potential difference is negative 70 millivolts on the inside at rest. So at rest, the voltage is negative 70, which means it is polarized. And that's how you're going to remember depolarization, repolarization, and hyperpolarization. So it's generated by the different concentrations of sodium, potassium, and chlorine. Chlorine's supposed to have a negative charge, so I kind of have a slanted negative there. So I'll try to correct that. But regardless, it's those three, and there's protein anions on the inside. For some reason, the negatives didn't come through. But the current flow is going to depend on that voltage. There's ionic differences due to the permeability of the neurolemma, which is the membrane, to sodium and potassium. And then there's also a sodium-potassium pump. That's the active transport pump that we talked about way, way back in chapter 3. And the reason for the sodium pump is that basically it restores the balance of the sodium in the extracellular fluid and the potassium in the intracellular fluid after all of those action potentials, which we'll be talking about coming up. So let's look at another view of the resting membrane potential, and there's a video of this that is going to help with that in mastering A&P. But the resting membrane potential depends on the differences in the sodium-potassium concentrations inside and out, and the permeability of the plasma membrane potentials. So normally the sodium should be about this amount. You don't need to know the amount, and there's a very small amount of potassium. But on the inside, there's a high amount of potassium and a low amount of sodium. This is showing the sodium-potassium pump, and that's what maintains this concentration after action potentials. But in a normal resting membrane, there's a permeability of sodium and potassium. The potassium loss is due to abundant leakage channels, which means that those leakage channels are very permeable. And if there were only potassium channels, the inside would be negative 90. But since there are sodium channels and not just potassium channels, when we add the potassium channels, we have a resting membrane potential of negative 70 millivolts. So now that we add the sodium potassium pump, that can restore the balance. So again, I want you to make sure that you remember in a normal cell, you've seen this before, we have our nucleus, but the inside is going to be negative compared to the outside. So we're just looking at it from a different view. So it's a comparison from the inside to the outside. So that phospholipid bilayer, that membrane, is going to look like a battery. It's going to have potassium on the inside, some amino acid protein anions, and sodium on the outside, chlorine on the outside. And the difference between the inside and the outside would be negative 70 millivolts compared to the outside. So when we measure either a graded potential or an action potential, we're measuring this number and what it changes to.